So it's great to be back and tell you about our most recent study. And this really comes out of an interest of my lab in learning and memory, what changes in your brain when you encode memories. But recently we've been very interested in what maintains memory. So why can you remember things for decades? And this is really an interesting question, especially because the brain really is this 3-pound sort of bowl of jelly. And so how do you construct something within that structure that will last for decades? It's not a silicon circuit. It's really very dynamic, as I'll talk about. But of course, we can all remember things way into our 70s and 80s from our childhood. And sometimes we don't. So we're interested in how do the normal mechanisms occur, but also how they go wrong in both age-related memory disorders, but also Alzheimer's disease and other diseases. So the brain, it consists of 100 billion neurons, and here we have a video, which I stole from the internet, which really looks at, sort of illustrates the dynamic function of the brain. So you can see these are millions of neurons located here, and they're firing away, they're communicating with each other, they form neuronal circuits, which underlies all our behavior. So what happens, we think, when you encode a new memory is that the connections between these neurons become stronger, and they form a new, novel, sort of unique neuronal circuit. And it's that circuit that actually is the memory, and you reactivate that circuit, you actually recall the memory. So this change in synaptic connectivity in the connections between neurons forming these circuits is what underlies learning. So to give you a little, I got to give you a little primer on synaptic transmission and plasticity and how this is involved. And so this is another schematic shown here. Here are just 2 neurons out of those billions. They have a connection point right here, which is called a synapse. And each of these neurons are connecting to about 10,000 other neurons. So there are quadrillions of these firing away in your brain right now, hopefully. And I'm hopefully going to change a couple of them. So what happens is if you blow up and look at the function, here's the contact point. There's a chemical release of neurotransmitters from this neuron that then impacts the release of the neurotransmitters shown here, and then binds to these neurotransmitter receptors on the postsynaptic side. So this is how they communicate. That's going on billions of a time every second in your brain. And, but this is not a static process. If it was just static, you were hardwired, you wouldn't learn anything, you wouldn't respond to your environment, you wouldn't have no way to adapt. So what happens is this synaptic connectivity changes in strength, and we think that's what underlies experience-dependent learning and encoding memories. So research over the last 20 years or so has really looked at this in detail, and I'm just going to tell you how it does it. So this is 20 to 30 years of work from my lab and many labs across the world, is that during learning, what happens is the synapse becomes stronger, and how that happens is that the receptors, neurotransmitter receptors, increase on the cell surface. So this is now a stronger synapse. So this recruitment of neurotransmitter receptors make a stronger synapse. This occurs throughout the brain in different pathways, and that's what physically encodes the memory. Okay? So the question is, how is this stable? Why, how can this last for minutes or hours, or decades? So we've been studying this for years, and we can look at the receptors. This is just looking at a receptor in a dish. This is a neuron we've grown in a dish, and all these little green spots are fluorescent receptors. So we use genetic techniques to make the receptors themselves actually glow green. This is adding GFP, green fluorescent protein. And so you can actually see them at all the synapses. These are the tens of thousands of synapses on this one neuron shown here. Now this is pretty cool, and we studied this for years. But recently we've been able to do this in an intact animal. So what we do is engineer a mouse so we can actually see the receptors and watch the receptors inside the mouse as it's learning something. So we can actually watch this process in the intact live mouse. So this is sort of an image we get. We're zooming down through the brain here, and we can image these neurons and the receptors. And then when we do a 3-dimensional projection of this, you can get an image like this. So this is an intact live animal while it's learning something. So it's pretty amazing. It's really a brand new technique. So we can actually observe the receptors. If we zoom in at the receptors, we can see the receptors. We can see the structure of the neuron with other genetic tricks. So you can see the synapse has lots of receptors in it. And you can actually watch this recruitment process. Remember I told you during learning, receptors get added to the synapse. So here's a synapse before learning, after learning. We can actually watch this in real time. This is a couple hours. But the remarkable thing is you can do this for days. So you can look at 30 days. You can look at what this receptor is here is the same as level as the receptors here. So this level of receptors is very stable. And this is bizarre because we know that the receptors are not stable. They turn over in the brain about every 4 or 5 days. So there's something unique. What is in this synapse that is stable? We know it's not the receptors that allows them to accumulate receptors and maintain the level of receptors at that synapse. So this comes to our newest study, and we studied protein degradation in synapses. As I said, most proteins last 1 to 7 days. But many of you know that some proteins, like crystalline in your eye, actually last a long time, last decades. And that's why people get cataracts, as I'll mention in a minute. That these proteins have been around for 70 years. They get oxidized, they get damaged, and they become cloudy. So we asked a simple question: are there stable proteins like crystalline or collagen in synapses? So I won't go through in the details, but what we did is we took mice and we labeled them with a heavy amino acid. So we feed them this food that costs about $50,000 for 10 mice, and then you switch them to normal chow with regular amino acids, light amino acids. And I won't go through it, but we can, doing this type of a study, we can determine the half-lives of proteins. So we isolate synapses and ask the question, are there synaptic proteins? Using proteomic techniques, we can analyze thousands of proteins. And we found there are certain proteins, like this protein, that don't turn over at all, while all the other proteins that we've been studying for years in the synapses do turn over very rapidly. And so it's this little bump here, these proteins here, about 50 proteins we estimate have half-lives of 1 year to 10 years. So this is a mouse, right? So this is a mouse that lives 2 years. So these proteins are incredibly stable. So now we're analyzing these proteins and trying to decide, determine, are they involved in learning and memory? Are they involved in long-term learning and memory? And also, what happens to them with age? And so you know what happens with cataracts. I was going to get some human pictures, but they look really weird, so I got a dog. So cataracts is one case. The other is, of course, osteoarthritis. So these are when these proteins that are long-lasting start to break down, you get oxidized, aggregate, you get these sort of diseases. So that's where we are, and this work, we're studying this work along with identifying a lot of other genes and proteins involved in learning and memory, but also schizophrenia, autism, Alzheimer's disease, and intellectual disability. So I'd be happy to take any questions, and here's the people who did the work and the funding. So thank you very much.
I'm Laura Carter from NIH. I thought your talk was interesting. But what happens if the receptors increase after the learning? Do they go back to some base level, or do they stay?
So after learning, they go up and they stay. So that's one of the really interesting things. We can, with this new technique, we can look over very long periods and watch, how long do they stay, how stable is the memory, and also how long do they stay. So they do stay a very long time, and that's the dilemma. Why does the cell know to add receptors constantly to that synapse only, not the other synapses?
And anything happen to make them go away? Like if they become
So
couch potatoes or something?
So we've studied memory erasure as well. So there are ways to actually reverse this potentiation and actually induce erasing memories. So you just Google Huganir erasing memories, you'll find out about that study.
Thank you.
I have a question over here. Corey Vancheri with Science News Magazine. Help me distinguish the learning part of things with the grabbing the memory when you need it to use the information. We've written about scientists who talk about the engram and trying to find that in the brain, and how do you distinguish that?
So, so we're very interested in that. We're trying to study that. Exactly what I'm trying to do is understand the engram. What is the code for memories? So the synaptic connections, we study the synapses, but that's part of a circuit which forms the memory, and that's the engram. So the learning is this process of adding the receptors, we think, is the majority of it. The stability is mediated by some unknown mechanism. Now, recalling memories is very interesting. How do you try to remember somebody's name and you can't remember their name, and usually people sort of search to the area, maybe, oh, I was here, 5 years ago at the press club, and maybe I remember her, or, and then it comes back. So it's, the way I think about it, and this is probably, I don't study this, but, you're sort of searching that space within your brain that's has related memories. And this is true that memories in time are linked in some ways. And so Alcino Silva has studied this at UCLA. And so once you get to that region, then you can, that name can sometimes pop into your mind. So I think it's re-engaging the same engram that has been formed during the learning process.
Thanks a lot.
Okay, thank you.